Subscribe to Boring Science for sleep for new videos every day. Now, let us begin. The organisms known as lichens have been observed growing upon stone surfaces across nearly every terrestrial environment on the planet Earth for a period of time that is estimated, through the examination of fossil evidence, to extend back approximately 400 million years into the geological past. A lichen is not, as was once commonly assumed by naturalists of earlier centuries, a single organism, but is rather a composite entity that arises from the stable and self-sustaining biological association between at least two, and in many cases 3 or more, distinct organisms belonging to entirely separate kingdoms of life. The primary component of this association is a fungus, which is referred to in the scientific literature as the mycobiont, and this fungal partner provides the structural framework of the lichen body, which is known as the thallus. The second essential component is a photosynthetic organism, which may be either a green alga belonging to the division Chlorophyta or a cyanobacterium, and this photosynthetic partner is referred to as the photobiont. In approximately 85% of all known lichen species, the photobiont is a green alga, while in approximately 10% of species, the photobiont is a cyanobacterium, and in the remaining species, both a green alga and a cyanobacterium are present simultaneously within the same thallus. The nature of the relationship between the mycobiont and the photobiont has been the subject of considerable scientific debate since the dual hypothesis of lichen biology was first proposed in the year 1867 by the Swiss botanist Simon Schwendener. Prior to Schwendener's revolutionary proposal, lichens had been classified as autonomous plants, and they were placed within the cryptogams alongside mosses and ferns by taxonomists who had no reason to suspect that what appeared to be a single organism was in fact a composite of 2 or more fundamentally different life forms. Schwendener's hypothesis, which he presented formally at a meeting of the Swiss Natural Science Society, was met with considerable skepticism and, in many quarters, outright hostility from the botanical establishment. The lichenologist William Nylander, who had spent decades cataloging and describing lichen species, rejected the dual hypothesis entirely and referred to Schwendener's supporters as followers of a destructive and unscientific ideology. It was not until the early 20th century, after the mycobiont and photobiont had been successfully separated and cultured independently in laboratory conditions, that the dual hypothesis gained widespread acceptance among the scientific community. The photobiont within a lichen thallus performs the function of photosynthesis, converting carbon dioxide and water into organic carbon compounds using energy derived from sunlight, and these organic compounds, primarily in the form of sugar alcohols such as ribitol and erythritol in the case of green algal photobionts, or glucose in the case of cyanobacterial photobionts, are transferred to the mycobiont. The mycobiont, in turn, provides the photobiont with a protected environment, shielding it from excessive ultraviolet radiation, desiccation, and mechanical damage. And the fungal hyphae also facilitate the absorption of water and dissolved mineral nutrients from the substrate upon which the lichen grows. This arrangement has been variously described as a mutualistic symbiosis, in which both partners derive benefit, or as a controlled parasitism, in which the mycobiont exploits the photobiont for nutritional gain while providing protection that the photobiont could, under certain environmental conditions, obtain through other means. The precise characterization of the relationship remains a matter of ongoing investigation and has not been definitively resolved by the scientific community as of the present time. The morphology of lichen thalia exhibits considerable variation across the approximately 20,000 species that have been described and catalogued by lichenologists worldwide. But the vast majority of these species can be classified into one of three principal growth forms, which are designated as crustose, foliose, and fruticose. The crustose growth form, which is exhibited by approximately 75% of all known lichen species, is characterized by a thallus that grows tightly oppressed to the substrate surface, forming what appears to be a thin crust or coating that cannot be removed from the substrate without also removing a portion of the substrate material itself. Crustose lichens are the growth form most commonly observed on stone surfaces, and their intimate physical contact with the rock substrate is a consequence of the fact that the lower surface of the thallus lacks a defined lower cortex and is instead composed of medullary hyphae that penetrate directly into the pores, cracks, and intergranular spaces of the stone. Some crustose lichens are described as endolithic, meaning that the majority of the thallus is actually embedded within the stone itself, with only the reproductive structures visible on the surface. The foliose growth form is characterized by a thallus that is leaf-like in appearance, with distinct upper and lower surfaces and lobes that extend outward from a central point of attachment. Foliose lichens are attached to the substrate by structures called rhizines, which are bundles of fungal hyphae that extend from the lower cortex and anchor the thallus to the rock, bark, or soil upon which it grows. Because foliose lichens are only loosely attached to their substrate, they can often be peeled away from the surface without significant damage to either the lichen or the substrate, a characteristic that distinguishes them from crustose species. The lobes of foliose lichens may range in width from less than 1 millimeter to several centimeters, and the thallus as a whole may achieve diameters of 10 to 30 centimeters or more over a period of many decades of growth. The fruticose growth form is characterized by a thallus that is shrub-like or hair-like in structure, with branches that may be either erect, growing upward from the substrate, or pendant, hanging downward from branches or rock overhangs. Fruticose lichens are attached to the substrate at a single basal point, and the branches are typically circular or flattened in cross-section. Well-known examples of fruticose lichens include the genus Usnea, commonly known as old man's beard, which forms long, pendant strands that may exceed 1 meter in length in undisturbed forest environments, and the genus Cladonia, which includes the reindeer lichens that form extensive ground cover in boreal and Arctic ecosystems. Fructose lichens are less commonly observed on stone surfaces than crustose or foliose species, though they do occur on rock faces and boulders in humid environments where moisture availability is sufficient to support their relatively high water requirements. In addition to these three principal growth forms, several intermediate or less common morphological types have been recognized by lichenologists. The squamulose growth form is characterized by small, scale-like lobes that are loosely attached to the substrate and may overlap one another. The leprose growth form consists of a powdery or granular thallus that lacks any organized internal structure and is composed entirely of loose clusters of fungal hyphae and algal cells. The filamentous growth form, which is relatively rare, consists of thread-like strands of fungal hyphae interwoven with chains of photobiont cells. These less common growth forms are of interest to specialists in lichen taxonomy, but are not generally significant in the context of lichen growth on stone surfaces, where the crustose and foliose forms predominate. The internal anatomy of a lichen thallus, when examined in cross-section under a light microscope, reveals a layered organization that is referred to as the stratified or heteromerous arrangement. In a typical foliose lichen, the uppermost layer is the upper cortex, which is composed of tightly packed fungal hyphae that are often pigmented and serve to protect the photobiont from excessive light exposure. Below the upper cortex lies the algal layer, also known as the photobiont layer, in which the cells of the green alga or cyanobacterium are distributed among loosely arranged fungal hyphae. Below the algal layer is the medulla, which is composed of loosely interwoven fungal hyphae with large air spaces between them, and this layer functions in gas exchange and water storage. The lowermost layer is the lower cortex, which is similar in structure to the upper cortex, but is often darker in color and gives rise to the rhizines by which the thallus is attached to the substrate. In crustose lichens, the lower cortex is typically absent, and the medullary hyphae extend directly into the substrate, creating an intimate physical and chemical interface between the lichen and the stone. The history of lichenology as a distinct scientific discipline may be traced back to the work of the French botanist Joseph Pitt-Ton de Tournefort, who in the year 1694 established the first systematic classification of lichens based on their external morphology. However, the study of lichens received its most significant early impetus from the work of the Swedish taxonomist Erik Akarius, who published a series of monumental works on lichen classification between the years 1803 and 1814, including the Methodus Lichenum in 1803 and the Lichenographia Universalis in 1810. Akarius is widely regarded as the father of lichenology and his classification system, though it has been extensively revised in the subsequent 2 centuries, established the foundation upon which all later taxonomic work on lichens has been built. Following Akarius, the Finnish lichenologist William Nylander dominated the field for much of the 19th century, describing thousands of new species and developing a classification system that was based primarily on the morphology of the reproductive structures, particularly the apothecia. which are the disc-shaped fruiting bodies in which the sexual spores of the mycobiont are produced. The water relations of lichens represent one of the most distinctive aspects of their physiology and are fundamentally different from those of vascular plants. Unlike vascular plants, which possess roots, vascular tissue, and stomata that allow them to absorb, transport, and regulate water within their tissues, lichens lack all of these structures and are entirely dependent on passive absorption of water from their external environment. When a dry lichen thallus is exposed to liquid water, either from rainfall, dew, fog, or snow melt, the thallus absorbs water rapidly through its entire surface, and the water content of the thallus may increase from less than 10% of its dry weight to between 150 and 300% of its dry weight within a period of minutes. This rapid rehydration activates the metabolic machinery of both the mycobiont and the photobiont, and photosynthesis, respiration, and growth resume within minutes to hours of wetting, depending on the species and the temperature. The rate at which the thallus subsequently loses water through evaporation is determined by the ambient temperature, humidity, and wind speed, and in exposed environments. A fully hydrated lichen thallus may return to a desiccated state within a few hours on a warm, dry day with moderate wind. The total number of hours per year during which a lichen thallus is sufficiently hydrated to support metabolic activity varies enormously across different climatic zones, from approximately 1,000 to 3,000 hours per year in humid maritime environments to as few as 100 to 500 hours per year in arid and semi-arid environments. And this variation in metabolically active time is one of the principal determinants of the wide range of growth rates observed among populations of the same species growing under different climatic conditions. The process by which a lichen becomes established on a stone surface is one that unfolds over a timescale that is measured not in days or weeks, but in months and years, and in some cases decades. The initial colonization of a bare stone surface typically begins with the arrival of fungal spores, which are dispersed by wind currents and may travel considerable distances from the parent thallus before settling on a suitable substrate. For a lichen to become established, the fungal spore must not only germinate successfully on the stone surface, but must also encounter a compatible photobiont partner within a sufficiently short period of time, as the germinating fungal hypha cannot survive for an extended period without a source of organic carbon. The probability of a successful encounter between a germinating mycobiont spore and a free-living photobiont cell on a bare stone surface is estimated to be extremely low, which partly explains why the initial colonization of newly exposed rock surfaces by lichens is such a slow process. Once a compatible mycobiont and photobiont have come into contact on the stone surface, the fungus begins to envelop the algal or cyanobacterial cells with its hyphae, and the earliest stages of thallus formation commence. In the case of crustose lichens, the developing thallus spreads outward across the stone surface as a thin, expanding disc, and the marginal hyphae at the advancing edge of the thallus extend into the microscopic irregularities of the stone surface, anchoring the lichen and beginning the slow process of biochemical interaction with the mineral substrate. The rate at which this initial establishment and early growth occurs is influenced by a complex interplay of environmental factors, including the availability of moisture, the intensity and duration of sunlight exposure, the ambient temperature regime, the chemical composition of the stone substrate, and the quality of the surrounding atmosphere. The stone surface itself presents both opportunities and challenges for the establishing lichen. The mineral composition of the rock determines the availability of essential nutrients such as calcium, magnesium, potassium, and iron, and the pH of the rock surface, which may range from strongly acidic in the case of granite and quartzite to strongly alkaline in the case of limestone and marble, influences which lichen species are able to colonize successfully. The physical texture of the stone, including its porosity, surface roughness, and the size and distribution of mineral grains, affects the ability of fungal hyphae to penetrate into the substrate and the capacity of the stone surface to retain moisture following rainfall or dew formation. Rough-textured, porous stones such as sandstone generally provide more favorable conditions for initial lichen colonization than smooth, dense stones such as polished marble or fine-grained basalt. As the greater surface area and water-holding capacity of porous stones enhance the survival of germinating spores and young thalli during periods of desiccation. The orientation of the stone surface with respect to sunlight and prevailing wind and rain patterns also exerts a significant influence on lichen establishment and subsequent growth. In the northern hemisphere, north-facing stone surfaces tend to support different lichen communities than south-facing surfaces, as the reduced direct sunlight on north-facing surfaces results in cooler temperatures and higher moisture retention, conditions that favor certain species of crustose and foliose lichens while inhibiting others. Similarly, stone surfaces that are sheltered from direct rainfall may support different lichen communities than exposed surfaces, as the absence of direct wetting may limit the establishment of species that require frequent hydration while favoring species that are adapted to obtaining moisture primarily from atmospheric humidity and dew. The ecological concept of primary succession, which describes the sequential colonization of a bare substrate by increasingly complex communities of organisms over time, is well illustrated by the colonization of newly exposed stone surfaces by lichens. In this context, lichens are generally regarded as pioneer organisms, meaning that they are among the first macroscopic life forms to become established on bare rock, and their presence initiates a cascade of ecological processes that gradually transform the stone surface into an environment capable of supporting more complex plant communities. The mechanical and chemical weathering of the stone by lichen hyphae and lichen-produced organic acids creates a thin layer of mineral particles and organic matter on the rock surface and this protosoil, which may accumulate at a rate of only a few hundredths of a millimeter per year, eventually becomes sufficient to support the germination of moss spores and, much later, the seeds of vascular plants. This process of soil formation on bare rock through biological weathering is known as pedogenesis, and it proceeds at a rate that is measured in centuries and millennia rather than in human lifetimes. The study of lichen colonization on stone surfaces has been greatly facilitated by the existence of dated stone structures, such as tombstones, buildings, walls, and monuments, which provide researchers with substrates of known age on which the rate of lichen colonization and growth can be measured directly. Churchyard tombstones have been particularly valuable in this regard, as they typically bear inscriptions that record the date of installation. They are composed of identifiable stone types, and they are distributed across a wide range of climatic conditions in regions where churchyards have been maintained for centuries. Studies conducted in churchyards across Europe and North America have documented the progressive colonization of tombstone surfaces by lichen communities over periods ranging from a few decades to several centuries. And these studies have provided foundational data for the development of lichenometric dating techniques, which will be discussed in greater detail in subsequent portions of this program. The diversity of lichen species that may be found growing on stone surfaces in any given location is influenced by a combination of local and regional factors, including the climate, the availability of different stone types, the presence or absence of atmospheric pollution, the history of land use in the surrounding area, and the composition of the regional lichen flora. In unpolluted environments with a variety of stone substrates, it is not uncommon to find 20 to 40 or more lichen species growing on stone surfaces within a relatively small area. And the specific composition of this lichen community provides information about the environmental conditions that prevail at that location. The use of lichen communities as biological indicators of environmental quality, particularly air quality, has been an active area of research since the mid-20th century and has provided valuable data for environmental monitoring programs in many countries. The reproduction of lichens occurs through both sexual and asexual mechanisms, and the relative importance of these two modes of reproduction varies among species and environmental conditions. Sexual reproduction in lichens is accomplished exclusively by the mycobiont, which produces spores in specialized fruiting structures that are visible on the surface of the thallus. The most common type of fruiting structure is the apothecium, which is a disc-shaped or cup-shaped structure, typically ranging from 0.5 to 5 millimeters in diameter, in which the spore-producing cells, known as asci, are arranged in a layer called the hymenium. Each ascus typically contains 8 spores, although the number may vary from 1 to over 100 depending on the species. The spores are released into the air when the ascus ruptures at maturity, and they are dispersed by wind to potentially great distances from the parent thallus. Upon landing on a suitable substrate, the spore germinates and produces a network of hyphae that must encounter a compatible photobiont partner in order for a new lichen thallus to develop. The probability of this encounter is sufficiently low that sexual reproduction is considered to be a relatively inefficient method of lichen propagation, and many lichen species supplement or replace sexual reproduction with various forms of asexual reproduction. The most common forms of asexual reproduction in lichens involve the dispersal of small fragments of the thallus that contain both the mycobiont and the photobiont, thereby eliminating the need for the two partners to find each other independently on the new substrate. Sordia are tiny granular structures, typically 25 to 100 micrometers in diameter, that consist of a few photobiont cells wrapped in a loose network of fungal hyphae. Sordia are produced in specialized structures called soralia, which are visible on the surface of the thallus as powdery or granular patches, and they are dispersed by wind, water, and animal contact. Isidia are small, cylindrical, or branched-out growths of the thallus surface that contain all the layers of the parent thallus, including the cortex, algal layer, and medulla, and that break off from the thallus and are dispersed to new substrates. Both Sordia and Isidia are capable of establishing new lichen thalli upon landing on a suitable substrate, and the success rate of establishment from these vegetative propagules is considerably higher than from sexual spores, as the propagule already contains a functional symbiotic association. The chemical ecology of lichens is a field of study that has revealed an extraordinary diversity of secondary metabolites produced by the mycobiont of lichen species. More than 1,000 distinct chemical compounds have been isolated from lichens, and the majority of these compounds are unique to lichens and are not found in any other group of organisms. These lichen substances, as they are collectively known, include depsides such as lechinoric acid and evernic acid, depsidones such as salazinic acid and norstitic acid, dibenzofurans such as usnic acid, and xanthones such as arthotheliin and lycoxanthone. The functions of these compounds within the lichen thallus are varied and include protection against ultraviolet radiation, deterrence of herbivory by invertebrates, inhibition of microbial infection, regulation of light transmission to the photobiont layer, and, as previously mentioned, the chelation of metal ions at the lichen-rock interface. Usnic acid, which is one of the most widely studied lichen substances, has been shown to possess antibiotic, antiviral, antifungal, and anti-inflammatory properties, and it has been investigated as a potential source of pharmaceutical compounds, although its toxicity to mammalian liver cells has limited its therapeutic applications. The global distribution of saxicolous lichens, which is the term used to describe lichens that grow on rock and stone surfaces, encompasses virtually every terrestrial biome, from tropical rainforests to polar deserts, and from sea-level coastal cliffs to alpine summits above 5,000 meters in elevation. Lichens have been recorded growing on stone surfaces at latitudes as extreme as 86 degrees south, within a few hundred kilometers of the geographic South Pole, and at altitudes exceeding 7,000 meters on peaks in the Himalayas, making them among the most widely distributed of all macroscopic terrestrial organisms. This extraordinary geographic range is a testament to the physiological adaptations that lichens have evolved, particularly their ability to tolerate extreme desiccation, extreme temperature fluctuations, and high levels of ultraviolet radiation, adaptations that allow them to thrive in environments where most other organisms cannot survive. The measurement of lichen growth rates has occupied the attention of researchers in the fields of lichenology, ecology, and geomorphology for well over a century. And the body of data that has accumulated during this period reveals that lichens are, as a general rule, among the slowest growing macroscopic organisms on the surface of the Earth. The radial growth rates of crustose lichens on stone surfaces have been measured in numerous studies conducted across a wide range of climatic zones and substrate types. And these measurements indicate that annual radial growth increments typically fall within a range of approximately 0.1 millimeters per year at the lower end to approximately 10 millimeters per year at the upper end, with the majority of species growing at rates between 0.5 and 3 millimeters per year. These figures represent the increase in radius of the roughly circular thallus measured from the center to the outer margin, and they should not be confused with the increase in diameter, which would be approximately twice the radial growth rate, assuming symmetrical expansion of the thallus in all directions. The specific growth rate exhibited by any individual lichen thallus is determined by a complex interaction of factors that include the species identity of the lichen, the age and size of the thallus, the chemical and physical properties of the stone substrate, the climatic conditions at the site, the orientation and inclination of the stone surface, the availability of atmospheric moisture, the quality of the surrounding air, and the degree of competition from neighboring lichenthali and other organisms. Among these factors, species identity is perhaps the most fundamental determinant of growth rate. As different lichen species possess inherently different metabolic capacities and growth potentials that are genetically determined and cannot be altered by environmental conditions. For example, the common crustose lichen Rhizocarpon geographicum, which is widely used in lichenometric dating studies, has been reported to grow at rates ranging from approximately 0.1 to 0.9 millimeters per year on stone surfaces in temperate and Arctic environments, while the foliose lichen Xanthoria parietina, which is commonly found on stone walls and buildings, has been reported to grow at rates of 1 to 3 millimeters per year under favorable conditions. The measurement of lichen growth rates on stone surfaces has been accomplished through several distinct methodological approaches, each of which possesses particular advantages and limitations. The most direct method involves the repeated measurement of identified individual lichenthali at fixed time intervals, typically using calipers, graduated rulers, or other precision measuring instruments to determine the diameter or radius of the thallus at each measurement point. This method, which is sometimes referred to as the direct measurement method, provides the most reliable data on growth rates, but requires that measurements be continued over a period of several years to several decades in order to obtain statistically meaningful results. As the annual growth increments of most crustose lichens are so small that they approach the limits of measurement precision over shorter time intervals. Studies employing the direct measurement method have been conducted at numerous locations worldwide, and some of the longest-running datasets extend over periods of 40 to 50 years or more of continuous monitoring. A second methodological approach to measuring lichen growth rates involves the use of fixed-point photography, in which high-resolution photographs of marked areas of stone surface are taken at regular intervals, typically annually, and the changes in lichen thallus size between successive photographs are measured using image analysis software. This photographic method has the advantage of providing a permanent visual record of lichen growth and colonization patterns over time, and it allows the simultaneous monitoring of multiple lichen thalli within a single photograph. The precision of photographic measurements has improved substantially with advances in digital imaging technology, and modern studies employing this method are capable of detecting growth increments of less than 0.1 millimeters per year. However, the photographic method is subject to potential sources of error arising from variations in camera position, lighting conditions, and the angle of view between successive photographs. And considerable care must be taken to ensure that these sources of error are minimized through the use of permanent photographic stations and standardized imaging protocols. A third approach to estimating lichen growth rates is based on the principle of lichenometry, which involves measuring the diameters of the largest lichen thalli growing on stone surfaces of known age, such as tombstones, building foundations, moraines deposited by glaciers at documented dates, or rock surfaces exposed by dated landslides or volcanic eruptions. By dividing the measured diameter of the largest lichen thallus by the known age of the substrate, an average growth rate can be calculated, and this rate can then be applied to estimate the age of stone surfaces of unknown age. This lichenometric approach has been widely employed in geomorphological studies, particularly in Arctic and alpine environments where other dating methods are difficult to apply, and it has provided valuable estimates of the ages of glacial moraines, rock glacier surfaces, and talus deposits. However, the accuracy of growth rate estimates derived from lichenometric measurements is limited by several sources of uncertainty, including the assumption that the largest thallus on a surface colonized the surface shortly after it was exposed, the assumption that the growth rate has remained constant over the entire period of growth, and the difficulty of accounting for periods of growth interruption or thallus mortality. The relationship between lichen thallus size and age is not a simple linear one, and extensive research has demonstrated that the growth rate of most lichen species varies systematically with the size and age of the thallus. This variation is most commonly described using the concept of the lichen growth curve, which plots the diameter or radius of the thallus as a function of time since establishment. The typical lichen growth curve consists of 3 distinct phases. The first phase, which is referred to as the colonization phase or the lag phase, encompasses the period immediately following initial establishment, during which the young thallus grows very slowly as it becomes established on the stone surface and develops the internal organization necessary for sustained growth. This phase may last for several years to several decades, depending on the species and environmental conditions, and the growth rate during this phase is typically well below the maximum rate that the species is capable of achieving. The second phase of the lichen growth curve is referred to as the Great Period of Growth, a term that was introduced by the Austrian lichenologist Roland Bestial in his pioneering work on lichenometric dating published in 1958. During the Great Period, the thallus exhibits its maximum rate of radial expansion, and the relationship between thallus diameter and time is approximately linear. The Great Period typically commences when the thallus has reached a diameter of approximately 10 to 20 millimeters and may continue for a period of several decades to several centuries, depending on the species and environmental conditions. It is during this phase that the growth rate is most consistent and predictable, and it is the growth rate during the Great Period that is most commonly used in lichenometric dating calculations. The third phase of the growth curve is characterized by a gradual decline in growth rate as the thallus approaches its maximum size, and this phase is sometimes referred to as the senescent phase or the declining phase. The reduction in growth rate during this phase is attributed to several factors, including the increasing metabolic cost of maintaining a large thallus, the decreasing ratio of actively growing marginal tissue to total thallus area, and the progressive deterioration of the central portions of the thallus through natural aging processes. In some species, the central portion of very old thalli may die and decompose, leaving a ring-shaped thallus that continues to expand at its outer margin while the inner portion degrades. A pattern of growth that has been observed in several species of rhizocarpon and other long-lived crustose lichens. The influence of moisture availability on lichen growth rates has been demonstrated in numerous studies conducted across a range of climatic conditions. Lichens are poikilohydric organisms, meaning that they lack the ability to regulate their internal water content and are entirely dependent on ambient moisture conditions for hydration. When a lichen thallus is dry, its metabolic activity ceases almost entirely, and photosynthesis, respiration, and growth are suspended until the thallus is re-wetted by rainfall, dew, fog, or atmospheric humidity. The amount of time during which a lichen thallus is in a hydrated and metabolically active state is a critical determinant of its annual growth rate, and studies have shown that lichens growing in humid environments with frequent fog or dew events tend to exhibit higher growth rates than lichens of the same species growing in arid environments, where the thallus is dry for extended periods. In the maritime climate of the British Isles, for example, where atmospheric humidity is consistently high and fog and drizzle are frequent, crustose lichens on stone surfaces have been reported to achieve growth rates that are approximately 50% higher than those of the same species growing on similar stone substrates in the drier continental climate of Central Europe. Temperature exerts its influence on lichen growth primarily through its effects on the rates of enzymatic reactions within the thallus, including the reactions of photosynthesis, respiration, and biosynthesis. Most lichen species exhibit optimal metabolic activity within a temperature range of approximately 10 to 25 degrees Celsius. Although many species are capable of net photosynthesis at temperatures as low as -5 to -10 degrees Celsius, particularly when the thallus is hydrated by snowmelt or by moisture from melting frost. The relationship between temperature and lichen growth rate is complicated by the fact that temperature also affects the rate of evaporation and therefore the duration of thallus hydration following wetting events. In warm environments, the rapid evaporation of moisture from the thallus surface may reduce the period of metabolic activity following a wetting event, potentially offsetting the positive effect of higher temperatures on enzymatic reaction rates. This interaction between temperature and moisture availability helps to explain why some of the highest lichen growth rates on stone surfaces have been recorded not in warm tropical environments, but in cool, humid maritime environments at temperate latitudes. The intensity and spectral composition of light reaching the lichen thallus influence growth primarily through their effects on the rate of photosynthesis by the photobiont. Most lichens are adapted to moderate light levels and may suffer photoinhibition, a reduction in photosynthetic efficiency caused by damage to the photosynthetic apparatus when exposed to very high light intensities. Conversely, lichens growing in deeply shaded locations may receive insufficient light to support rates of photosynthesis adequate for sustained growth, and the thallus may gradually decline and eventually die if light levels fall below the compensation point at which photosynthetic carbon fixation equals respiratory carbon loss. The optimal light environment for lichen growth varies considerably among species, and this variation is reflected in the distribution of different lichen species across stone surfaces with different degrees of sun exposure. Sun-loving or heliophilous species, such as many species of Caloplaca and Xanthoria, tend to colonize stone surfaces that receive direct sunlight for much of the day, while shade-tolerant or sciophilous species, such as many species of Lepraria and Opegrapha, are found on stone surfaces in shaded locations such as the undersides of overhanging rocks or the north-facing walls of buildings. The chemical composition of the stone substrate upon which a lichen grows influences growth through several mechanisms. The mineral nutrients released by the slow weathering of the stone surface provide essential elements for lichen metabolism, including calcium, magnesium, potassium, iron, manganese, and phosphorus. And the availability of these elements is determined by the mineralogical composition of the rock. Calcareous stones such as limestone and marble, which are composed predominantly of calcium carbonate, provide abundant calcium, but may be deficient in other elements such as iron and manganese, while siliceous stones such as granite and quartzite provide a different balance of mineral nutrients. The pH of the stone surface also influences lichen growth, as the metabolic processes of both the mycobiont and the photobiont are sensitive to hydrogen ion concentration. Most lichen species can be classified as either calcicole, preferring alkaline substrates such as limestone, or calcifuge, preferring acidic substrates such as granite. And this substrate preference is a fundamental aspect of lichen ecology that determines the composition of lichen communities on different stone types. The quality of the atmosphere surrounding a lichen thallus has been recognized as a significant determinant of lichen growth and survival since the early observations of the Finnish botanist William Nylander, who noted in 1866 that lichens were disappearing from trees and stones in the environs of Paris as industrial pollution increased. Atmospheric pollutants, particularly sulfur dioxide, nitrogen oxides, and heavy metals, have been shown to have strongly deleterious effects on lichen growth. And many lichen species are incapable of surviving in environments where the concentration of sulfur dioxide in the ambient air exceeds approximately 30 to 40 micrograms per cubic meter on an annual average basis. The sensitivity of lichens to air pollution is attributed to several factors, including their lack of a protective cuticle or epidermis, which allows pollutants to be absorbed directly into the thallus from rainwater and atmospheric moisture. Their inability to shed damaged tissues and replace them with new growth in the manner of vascular plants, and the sensitivity of the photobiont, particularly green algal photobionts, to acidification of the thallus caused by the absorption of sulfur dioxide and its conversion to sulfurous and sulfuric acid within the lichen tissues. The competitive interactions between neighboring lichen thalli on a stone surface also influence the growth rates of individual thalli, as the expansion of one thallus may be impeded by physical contact with an adjacent thallus. When two expanding lichen thalli come into contact at their margins, one of several outcomes may occur, depending on the species involved and the relative vigor of the two thalli. In some cases, the two thalli may form a distinct boundary line where growth is mutually inhibited, resulting in a sharp demarcation between the two colonies. In other cases, one thallus may overgrow the margin of the other, gradually displacing the subordinate colony from the contested area. These competitive interactions can significantly reduce the effective growth rate of individual thalli in densely colonized areas, where the available stone surface has been completely partitioned among neighboring colonies, and further expansion is possible only through the displacement of competitors. Studies of lichen competition on stone surfaces have shown that the outcome of competitive interactions is influenced by the species identity of the competitors, the relative sizes of the thalli, and the environmental conditions at the site, with certain species consistently dominant over others in competitive encounters. The seasonal pattern of lichen growth on stone surfaces has been investigated in several long-term studies that have employed continuous or frequent monitoring of thallus dimensions throughout the annual cycle. These studies have generally found that lichen growth is not uniform throughout the year, but rather exhibits a seasonal pattern that is correlated with the prevailing climatic conditions. In temperate regions of the northern hemisphere, most crustose lichens on stone surfaces exhibit their maximum growth rates during the spring and autumn months, when temperatures are moderate and moisture availability is relatively high, while growth rates are reduced during the summer months, when high temperatures and low humidity may cause extended periods of thallus desiccation, and during the winter months, when low temperatures reduce metabolic activity even when the thallus is hydrated. In Arctic and alpine environments, Lichen growth is confined primarily to the brief summer growing season, during which temperatures are above freezing and moisture is available from snowmelt and precipitation, and the annual growth increment is correspondingly small. The relationship between altitude and lichen growth rates on stone surfaces has been documented in studies conducted along altitudinal gradients in mountain ranges on several continents. These studies have generally found that lichen growth rates decrease with increasing altitude, reflecting the combined effects of lower temperatures, shorter growing seasons, increased ultraviolet radiation, and, in some mountain ranges, reduced precipitation at higher elevations. On Mount Kenya in equatorial East Africa, for example, the growth rate of Rhizocarpon geographicum has been found to decrease from approximately 0.5 millimeters per year at an altitude of 3,500 meters to approximately 0.1 millimeters per year at an altitude of 4,500 meters, a five-fold reduction over an altitudinal range of only 1,000 meters. Similar altitudinal gradients in lichen growth rates have been documented in the European Alps, the Scandinavian mountains, the Rocky Mountains of North America, and the Southern Alps of New Zealand. And these gradients have important implications for the accuracy of lichenometric dating in mountain environments, where growth rate calibrations obtained at one altitude may not be applicable to sites at different elevations. The size-growth rate relationship in lichens has been further investigated through statistical analyses of large datasets compiled from multiple study sites and spanning multiple decades of observation. These analyses have confirmed that the relationship between thallus diameter and annual growth rate follows a characteristic pattern that can be modeled using a variety of mathematical functions, including logarithmic, polynomial, and asymptotic growth models. The choice of mathematical model used to describe the growth curve has significant implications for the age estimates derived from lichenometric dating, and the selection of an inappropriate model can introduce systematic errors of 50% or more into the estimated ages of stone surfaces. For this reason, considerable effort has been devoted to the development and validation of growth curve models that accurately represent the observed patterns of lichen growth across the full range of thallus sizes, from the smallest detectable thalli of 1 to 2 millimeters in diameter to the largest thalli exceeding several hundred millimeters in diameter. The effect of latitude on lichen growth rates is closely related to the effect of altitude, as both increasing latitude and increasing altitude are associated with decreasing mean annual temperature and shorter growing seasons. Studies comparing lichen growth rates across latitudinal gradients have demonstrated that the radial growth rate of Rhizocarpon geographicum decreases from approximately 0.8 to 1.0 millimeters per year at latitudes of 45 to 50 degrees north, where the climate is relatively mild and the growing season extends for 6 to 8 months, to approximately 0.1 to 0.3 millimeters per year at latitudes of 70 to 75 degrees north, where the growing season is limited to 2 to 4 months per year and winter temperatures are extremely low. At the extreme southern latitudes of the Antarctic Peninsula, growth rates of Rhizocarpon geographicum as low as 0.07 millimeters per year have been reported. And at inland Antarctic locations with even more extreme climatic conditions, growth rates may be lower still, although the scarcity of long-term monitoring data from these remote locations makes precise estimates difficult to obtain. The influence of aspect, or the compass direction in which a stone surface faces, on lichen growth rates has been documented in several detailed studies conducted at individual sites where other environmental variables could be held approximately constant. In the northern hemisphere, south-facing stone surfaces receive more direct solar radiation than north-facing surfaces, and this difference in insolation results in higher surface temperatures and more rapid drying of the stone surface following wetting events. The net effect of these microclimatic differences on lichen growth rates depends on the relative importance of temperature and moisture as limiting factors for lichen metabolism at the study site. In cool, humid climates where moisture is rarely limiting, south-facing surfaces may support higher lichen growth rates due to the warmer temperatures, while in warm, dry climates where moisture is the primary limiting factor, north-facing surfaces may support higher growth rates due to the longer periods of thallus hydration. Studies conducted in the maritime climate of western Norway have found that the growth rate of Rhizocarpon geographicum on south-facing granite surfaces was approximately 20% higher than on north-facing surfaces of the same rock type, while studies in the semi-arid climate of the interior western United States have found the opposite pattern, with growth rates on north-facing surfaces exceeding those on south-facing surfaces by a similar margin. The phenomenon of lichen dormancy, which is the complete cessation of metabolic activity during periods of unfavorable environmental conditions, is a fundamental aspect of lichen physiology that has profound implications for the interpretation of lichen growth rates. When a lichen thallus becomes desiccated, its water content may fall below 10% of its dry weight, and at this level of dehydration, all metabolic processes, including photosynthesis, respiration, and growth, cease entirely. The thallus enters a state of suspended animation that may persist for days, weeks, months, or even years without causing permanent damage to the organism. Laboratory experiments have demonstrated that dried lichen thalli can be stored for periods of several years and will resume normal metabolic activity within minutes to hours of being rewetted, a capacity for desiccation tolerance that is shared by very few other groups of organisms. This ability to tolerate prolonged desiccation is attributed to the production of protective compounds such as trehalose and other sugars that stabilize cell membranes and proteins during dehydration, and to the structural properties of the thallus that minimize mechanical damage during the contraction that accompanies water loss. The behavior of lichen growth on different types of stone has been the subject of extensive investigation by researchers in the fields of geomorphology, biogeochemistry, and conservation science, and the accumulated body of evidence demonstrates that the mineralogical composition, texture, porosity and surface chemistry of the stone substrate exert profound influences on the rate, pattern, and species composition of lichen colonization. Granite, which is a coarse-grained igneous rock composed primarily of the minerals quartz, feldspar, and mica, is one of the most commonly studied substrates for lichen growth. Owing both to its widespread occurrence as a building stone and monument material and to its prevalence as a natural rock surface in glaciated and mountainous landscapes. The surface of granite provides a slightly acidic substrate with a pH typically ranging from approximately 5.5 to 6.5. And this acidic character makes granite a preferred substrate for calcifuge lichen species such as Rhizocarpon geographicum, Licidia auriculata, and various species of the genus Porphyria. The coarse-grained texture of granite, with individual mineral crystals typically ranging from 1 to 5 millimeters in diameter, provides a rough surface with numerous microscopic crevices and pits between mineral grains. And these surface irregularities facilitate the initial attachment of lichen propagules and the subsequent penetration of fungal hyphae into the stone. Studies of lichen growth rates on granite surfaces have yielded a range of values that vary with species, climate, and geographic location. In the cool maritime climate of southern Norway, the radial growth rate of Rhizocarpon geographicum on granite boulders and outcrops has been estimated at approximately 0.3 to 0.5 millimeters per year during the Great Period of Growth, based on measurements of thalli growing on surfaces of known age. In the warmer maritime climate of Western Ireland, growth rates of the same species on granite have been estimated at approximately 0.5 to 0.8 millimeters per year, reflecting the longer growing season and higher average moisture availability at these lower latitude sites. In the continental climate of the interior of North America, where temperature extremes are greater and annual precipitation is lower, growth rates of Rhizocarpon geographicum on granite have been estimated at approximately 0.1 to 0.3 millimeters per year, values that are among the lowest reported for this species in the published literature. Limestone, which is a sedimentary rock composed predominantly of the mineral calcite, with the chemical formula calcium carbonate, presents a markedly different substrate for lichen growth compared to granite. The surface of limestone is alkaline, with a pH typically ranging from approximately 7.5 to 8.5, and this alkaline character favors the establishment of calcicoal lichen species such as Varicaria nigrescens, Caloplaca flavescens, Aspercilia calcarea, and various species of the genus Lecanora. The texture of limestone varies considerably depending on its geological origin and subsequent diagenetic history, ranging from the extremely fine-grained texture of lithographic limestone, in which individual mineral particles are less than 4 micrometers in diameter, to the coarse-grained texture of bioclastic limestone, which is composed of fragments of shells, corals, and other calcareous organisms cemented together by a matrix of crystalline calcite. The porosity of limestone also varies widely, from less than 1% in dense crystalline varieties to more than 20% in some bioclastic and oolitic varieties. And this variation in porosity affects both the water-holding capacity of the stone and the ease with which lichen hyphae can penetrate into the substrate. The growth rates of lichens on limestone surfaces tend to be somewhat higher, on average, than those on granite surfaces in the same geographic location. A difference that is attributed to the greater availability of calcium and other mineral nutrients from the limestone substrate and to the generally higher water-holding capacity of limestone compared to granite. Studies conducted in the limestone regions of the English Pennines have reported radial growth rates of Aspergillia calcarea on limestone surfaces of approximately 0.6 to 1.0 millimeters per year, while growth rates of calcicoal species such as Caloplaca flavescens on limestone tombstones in English churchyards have been estimated at approximately 1.0 to 2.0 millimeters per year. However, the higher dissolution rate of limestone compared to granite means that lichen on limestone surfaces may be subject to undermining by chemical dissolution of the substrate beneath the thallus, a process that can lead to the detachment and loss of portions of the thallus and thereby reduce the apparent long-term growth rate. Sandstone, which is a sedimentary rock composed of sand-sized mineral grains, predominantly quartz, cemented together by a matrix of silica, calcite, iron oxide, or clay minerals, provides a substrate for lichen growth that combines characteristics of both granite and limestone, depending on the nature of the cementing matrix. Silica-cemented sandstones have an acidic surface chemistry similar to granite and tend to support calcifuge lichen communities, while calcite-cemented sandstones have an alkaline surface chemistry similar to limestone and tend to support calcicoal communities. The most distinctive feature of sandstone as a lichen substrate is its typically high porosity, which commonly ranges from 5 to 25% and may exceed 30% in some varieties. This high porosity gives sandstone an exceptional capacity to absorb and retain water, and the slow release of moisture from the interior of the stone following rainfall or dew events prolongs the period during which the lichen thallus remains hydrated and metabolically active. As a consequence, lichen growth rates on porous sandstone surfaces may be higher than on less porous stone types in the same environment, particularly in climates where moisture availability is a limiting factor for lichen growth. Basalt, which is a fine-grained igneous rock of dark color composed primarily of the minerals plagioclase, pyroxene, and olivine, provides a substrate for lichen growth that is characterized by a slightly alkaline to neutral surface chemistry, with a pH typically ranging from approximately 6.5 to 8.0. The fine-grained texture of basalt, with individual mineral crystals typically less than 1 millimeter in diameter, presents a relatively smooth surface that is less favorable for initial lichen colonization than the rougher surfaces of granite and sandstone, and the rate of lichen colonization on freshly exposed basalt surfaces is generally slower than on these coarser-grained rock types. However, once established, lichens on basalt surfaces may achieve growth rates comparable to those on granite. and the relatively high content of iron and magnesium in basalt provides essential mineral nutrients that may support robust lichen growth over the long term. Studies of lichen growth on basalt lava flows of known age in Iceland and Hawaii have provided important data for lichenometric dating in volcanic environments, with growth rates of Rhizocarpon geographicum on basalt in Iceland estimated at approximately 0.3 to 0.5 millimeters per year. Marble, which is a metamorphic rock formed by the recrystallization of limestone under conditions of elevated temperature and pressure, provides a substrate for lichen growth that is chemically similar to limestone but physically distinct. The crystalline texture of marble, with interlocking calcite crystals that may range from less than 1 millimeter to several centimeters in diameter, produces a surface that is often smoother and less porous than that of unmetamorphosed limestone. And this reduced surface roughness and porosity may slow the initial colonization of marble surfaces by lichens compared to limestone. The chemical composition of marble, being predominantly calcium carbonate with minor amounts of other minerals, favors the same calcical lichen species that are found on limestone. And the lichen communities on marble monuments and buildings are generally similar in species composition to those on limestone structures in the same geographic area. The use of marble as a prestige building material and monument stone throughout the history of Western civilization has resulted in the existence of numerous marble structures of known age, from ancient Greek temples to modern cemetery headstones. And these structures have provided valuable substrates for studies of lichen colonization and growth over periods ranging from decades to millennia. The chemical weathering of stone by lichens is a process that has been studied extensively since the early 20th century, and it is now well established that lichens contribute significantly to the breakdown of rock surfaces through the production of organic acids and other biochemically active compounds. The most important of these compounds is oxalic acid, which is produced by the mycobiont of many lichen species and is excreted at the interface between the lichen thallus and the stone substrate. Oxalic acid reacts with the calcium, magnesium, and iron in the rock minerals to form metal oxalate crystals, which have been detected at the lichen rock interface using techniques such as X-ray diffraction, scanning electron microscopy, and Fourier transform infrared spectroscopy. On limestone and marble surfaces, the reaction of oxalic acid with calcium carbonate produces calcium oxalate in two mineral forms, woolite, which has the formula calcium oxalate monohydrate, and wetolite, which has the formula calcium oxalate dihydrate, and these calcium oxalate crystals accumulate at the lichen rock interface as a thin layer that may be several tens of micrometers in thickness. In addition to oxalic acid, lichens produce a diverse array of other organic compounds, collectively known as lichen substances or lichen secondary metabolites, many of which have the capacity to chelate metal ions and thereby accelerate the dissolution of rock minerals. More than 1,000 distinct lichen substances have been identified through chemical analysis, and these compounds, which include depsides, depsidones, dibenzofurans, xanthones, and fulvinic acid derivatives, are produced by the mycobiont and are deposited on the surfaces of the fungal hyphae within the thallus. The role of these lichen substances in rock weathering is complex and not fully understood, but experimental studies have demonstrated that extracts containing lichen substances can increase the dissolution rate of common rock-forming minerals, such as feldspar, quartz, and calcite by factors ranging from approximately 2 to 10 times the dissolution rate in the absence of lichen substances. The physical weathering of stone by lichens occurs through the mechanical action of fungal hyphae that penetrate into the substrate along grain boundaries, cleavage planes, and microfractures in the rock. The hyphae of crustose lichens may penetrate to depths of several millimeters below the stone surface, and as these hyphae grow and expand, they exert physical pressure on the surrounding mineral grains, gradually prying them apart and loosening the structure of the rock. This process of hyphal penetration and grain separation is enhanced by the repeated wetting and drying of the lichen thallus, which causes cyclical swelling and contraction of the hyphae and the surrounding rock matrix, generating mechanical stresses that contribute to the progressive disintegration of the stone surface. The combined effect of chemical and physical weathering by lichens has been estimated to produce a rate of surface lowering on granite of approximately 0.1 to 1.0 millimeters per century, and on limestone of approximately 1 to 5 millimeters per century, values that, while small on a human timescale, represent a significant geomorphological process when considered over periods of thousands to millions of years. The use of lichen growth as a tool for dating stone surfaces, a technique known as lichenometry, was developed into a systematic methodology by the Austrian botanist Roland Bechtel, who published his foundational work on the subject in 1950 and 1958. Bechtel's approach was based on the premise that the diameter of the largest lichen thallus on a stone surface could be used as a proxy for the age of that surface, provided that the relationship between thallus diameter and age was known for the lichen species and geographic region in question. To establish this relationship, Bechtel measured the diameters of the largest thalli of Rhizocarpon geographicum growing on stone surfaces of known age, including tombstones, building foundations, mine tailings, and moraines deposited by glaciers at historically documented dates, and he used these measurements to construct a growth curve that could then be applied to surfaces of unknown age. Bechtel's lichenometric method was quickly adopted by geomorphologists working in Arctic and alpine environments, where it provided a means of dating glacial landforms that could not be dated by other methods. In these environments, the retreat of glaciers exposes fresh rock surfaces that are progressively colonized by lichens, and the size of the largest lichen thalli on a moraine provides an estimate of the time elapsed since the glacier deposited that moraine and retreated from it. Lichenometric dating has been applied to glacial moraines in numerous mountain ranges and polar regions, including the Alps, the Scandinavian mountains, the Rocky Mountains, the Andes, the Himalayas, Iceland, Svalbard, Greenland, and Antarctica. and the technique has provided age estimates for landforms ranging from a few decades to several thousand years old. In favorable conditions, lichenometric dates are considered to be accurate to within approximately 10 to 20% of the true age. Although the precision of the method decreases for older surfaces due to the declining growth rate of senescent thalli and the increasing probability that the original colonizing thallus has been lost through natural mortality. The application of lichenometry in archaeological contexts has provided valuable information about the ages of stone structures, ruins, and artifacts in situations where other dating methods are unavailable or impractical. Lichenometric dates have been obtained for stone walls, cairns, and other structures constructed by past human populations in regions as diverse as the Scottish Highlands, the Mediterranean Basin, the Andes of South America, and the islands of the Arctic. In these applications, the size of the largest lichen thallus on a stone that was placed in its current position by human activity provides an estimate of the minimum time elapsed since the structure was constructed or last modified. However, the interpretation of lichenometric dates in archaeological contexts requires careful consideration of the possibility that the stones used in construction may have been previously colonized by lichens before they were incorporated into the structure, a circumstance that would result in an overestimate of the age of the construction. The accuracy and reliability of lichenometric dating have been the subject of ongoing debate within the scientific community, and several limitations of the method have been identified by researchers who have attempted to validate lichenometric dates against independent dating methods. One fundamental limitation is the assumption that the largest thallus on a surface represents the first colonizer, whereas in reality, there may have been a significant time lag between the exposure of the surface and the arrival of the first lichen propagule. This colonization lag has been estimated to range from as little as 5 years in humid lowland environments to as much as 30 years or more in arid or high-altitude environments, and failure to account for this lag results in an underestimate of the true age of the surface. A second limitation is the assumption that the growth rate of the lichen species used for dating has remained constant over the entire period of growth, whereas in fact, growth rates may have varied in response to climatic fluctuations over the centuries or millennia during which the thallus was growing. A third limitation is the difficulty of accurately measuring the diameter of large, irregularly shaped lichen thalli, particularly those that have merged with neighboring thalli or that have lost portions of the thallus through natural mortality or disturbance. Despite these limitations, lichenometry remains a valuable dating tool in geomorphological and archaeological research, particularly in environments where other dating methods are not applicable or are prohibitively expensive. The method has been refined and improved since Bestial's original work through the development of more sophisticated statistical approaches to growth curve construction, the use of multiple lichen species for cross-validation, and the calibration of growth curves against independent dating methods such as radiocarbon dating, dendrochronology, and historical records. The application of digital image analysis techniques to the measurement of lichen thallus dimensions has also improved the precision and reproducibility of lichenometric measurements, and the use of repeat photography and satellite imagery has enabled the monitoring of lichen growth over large spatial scales and long time periods that would not be feasible through ground-based measurements alone. The specific interaction between lichen biochemistry and different stone mineralogies has been investigated through laboratory dissolution experiments in which polished stone surfaces have been incubated with lichen thalli or with solutions containing lichen-derived organic acids under controlled conditions. These experiments have demonstrated that the rate of mineral dissolution beneath a lichen thallus is typically 2 to 5 times greater than the rate of abiotic dissolution of the same mineral in the absence of lichen-derived compounds, and that the degree of enhancement varies with both the mineral species and the composition of the lichen-derived acids. Feldspar minerals, which are the most abundant mineral group in the Earth's crust and a major component of granite and other igneous rocks, are particularly susceptible to lichen-mediated dissolution, as the aluminum and alkali metal ions in feldspar form stable complexes with oxalic acid and other lichen-produced chelating agents. The dissolution of feldspar by lichen acids produces a residue of clay minerals, primarily kaolinite and alite, which accumulates at the lichen-rock interface and contributes to the development of a thin protosoil layer between the lichen thallus and the unweathered rock beneath. The formation of calcium oxalate patinas on limestone and marble surfaces by lichen activity is a phenomenon that has attracted considerable attention from conservation scientists, as these mineral patinas may play a protective role by forming a relatively insoluble coating over the more soluble calcium carbonate substrate. Studies conducted on marble monuments in Italy have found that surfaces bearing well-developed calcium oxalate patinas, which are often associated with former or current lichen colonization, exhibit lower rates of surface recession due to acid rain dissolution than unprotected surfaces lacking such patinas. This finding has led some researchers to suggest that the controlled encouragement of lichen colonization on limestone and marble monuments might actually contribute to the long-term preservation of these structures. Although this view remains controversial and is not universally accepted within the conservation community, the weathering of stone surfaces by lichens also involves the physical disruption of the mineral fabric through the expansion and contraction of lichen thalli in response to cyclical wetting and drying. When a dry lichen thallus absorbs water, it may expand in volume by as much as 30 to 50% within a period of minutes, and this rapid expansion generates mechanical stresses at the interface between the thallus and the stone surface that can cause the detachment of individual mineral grains and the widening of microfractures in the rock. The magnitude of these mechanically induced stresses has been estimated through direct measurement using strain gauges attached to stone surfaces beneath lichen thalli, and the results indicate that the pressures generated by thallus expansion can exceed 1 megapascal, which is sufficient to cause the failure of weakly cemented stone materials, such as poorly consolidated sandstone and weathered limestone. Over repeated cycles of wetting and drying, the cumulative effect of these mechanical stresses contributes significantly to the progressive disintegration of the stone surface, a process that is sometimes referred to as biophysical weathering to distinguish it from the biochemical weathering caused by lichen-produced acids and chelating agents. The ecological succession of lichen communities on stone surfaces is a process that unfolds over timescales ranging from decades to centuries, and the sequential changes in species composition that characterize this succession have been documented through long-term observational studies conducted in numerous geographic regions. The initial colonization of a bare stone surface is typically accomplished by a small number of pioneer species that are adapted to the harsh conditions of the uncolonized substrate, including intense solar radiation, rapid temperature fluctuations, and limited moisture and nutrient availability. On acidic stone surfaces such as granite, the pioneer community is commonly dominated by crustose species belonging to the general Acidia, Porphyria, and Rhizocarpon, while on alkaline stone surfaces such as limestone, the pioneer community typically includes crustose species of the genera Varicaria, Aspersilia, and Caloplaca. These pioneer species are generally characterized by slow growth rates, high tolerance of environmental extremes, and the ability to establish and persist on substrates with minimal organic matter content. As the pioneer lichen community becomes established and the processes of biological weathering begin to modify the stone surface, conditions gradually become more favorable for the colonization of additional lichen species, and the diversity of the lichen community increases over time. The accumulation of organic matter from dead and decaying lichen tissue, the trapping of wind-blown dust and soil particles by the rough surface of the lichen thalli, and the modification of the microclimate at the stone surface through the shading and moisture-retaining effects of the lichen cover all contribute to the creation of a more hospitable environment for later successional species. Foliose lichens, which generally require a somewhat more developed substrate than crustose species, typically appear on the stone surface several decades to a century after the initial colonization by crustose pioneers and their larger, more complex thalli further modify the surface environment by increasing the capacity for water retention and organic matter accumulation. The successional trajectory of lichen communities on stone surfaces is influenced by the same suite of environmental factors that determine the growth rates of individual lichen species, including climate, substrate chemistry, surface orientation, and atmospheric quality. In polluted environments, the successional sequence may be truncated or altered, as pollution-sensitive species are unable to establish even when other conditions are favorable, and the lichen community may remain dominated by a small number of pollution-tolerant species indefinitely. In unpolluted environments with favorable climatic conditions, the lichen community on a stone surface may continue to develop and increase in species diversity for several centuries, eventually reaching a relatively stable composition that changes only slowly over time and that is referred to as the climax community. The sensitivity of lichens to atmospheric pollution, particularly to sulfur dioxide and nitrogen oxides produced by the combustion of fossil fuels, has made them one of the most widely used groups of organisms for biological monitoring of air quality. The relationship between lichen distribution and air pollution was first noted systematically by William Nylander in Paris in 1866, and since that time, numerous studies have documented the decline and disappearance of lichen species in urban and industrial areas where pollution levels exceed the tolerance thresholds of sensitive species. The concept of lichen desert, a term used to describe the zone immediately surrounding a major pollution source in which no lichens can survive, was introduced in the mid-20th century and became a powerful tool for visualizing the spatial extent of air pollution around cities and industrial complexes. In many European cities during the 1950s and 1960s, the lichen desert extended for distances of 10 to 30 kilometers from the city center, and the progressive recolonization of these areas by lichens following the implementation of clean air legislation has provided dramatic evidence of the effectiveness of pollution control measures. The use of lichens as indicators of air quality has been formalized through the development of standardized monitoring protocols, including the Index of Atmospheric Purity, which was introduced by de Slover and LeBlanc in 1968. This index is calculated from the number and abundance of lichen species present on standard substrate trees at monitoring sites, and higher values of the index indicate better air quality. Variations and refinements of this index have been developed and applied in monitoring programs in numerous countries, and the resulting data have been used to produce maps of air quality that complement the data obtained from instrumental monitoring networks. In the context of lichen growth on stone surfaces, the effects of air pollution are manifested not only in the reduction of species diversity, but also in the reduction of growth rates of surviving species, and in the visible damage to lichen thalli caused by the absorption of acidic pollutants and heavy metals. The stone surfaces of famous monuments and buildings around the world support lichen communities that have, in many cases, been growing for centuries. And the relationship between these lichen communities and the preservation of the stone substrate has been a subject of both scientific interest and practical concern. The megalithic monuments of Stonehenge in southern England, which were erected approximately 4,500 years ago, support diverse lichen communities on their stone surfaces, and the lichens growing on these stones have been the subject of detailed surveys that have recorded more than 75 species of lichen on the monument. The question of whether these lichens should be removed to protect the stone from biological weathering or left in place as an integral part of the monument's heritage has been debated by conservators and heritage managers for decades, and the current management approach favors the retention of the lichen communities on the grounds that their removal would cause more damage to the stone surfaces than their continued presence. Similar debates about the management of lichen communities on historic stone structures have occurred in relation to many other famous monuments and buildings, including the Parthenon in Athens, the medieval cathedrals of Europe, the Moai statues of Easter Island, the ancient temples of Angkor Wat in Cambodia, and the rock-cut churches of Lalibela in Ethiopia. In each of these cases, the decision about whether to remove or retain the lichen communities involves a balancing of the potential damage caused by lichen-mediated weathering against the potential damage caused by removal methods, the aesthetic and ecological value of the lichen communities themselves, and the recognition that some degree of biological weathering is an inevitable and natural process that cannot be entirely prevented without enclosing the monument in a controlled environment. The development of non-destructive methods for controlling lichen growth on stone monuments, such as the application of biocides that kill the lichen without requiring physical scraping or scrubbing of the stone surface, has provided conservators with additional options for managing lichen communities on heritage structures, although the long-term effects of these biocides on the stone and on the broader ecology of the monument are not yet fully understood. The global biodiversity of lichens is remarkable, with current estimates suggesting that there are approximately 20,000 described species worldwide, and the true number of species, including those not yet discovered and described by taxonomists, is believed to be considerably higher, with some estimates suggesting a total global lichen flora of 28,000 or more species. This diversity is distributed across approximately 115 families and nearly 1,000 genera of lichenized fungi, and it encompasses an extraordinary range of morphological, chemical, and ecological variation. The hotspots of lichen biodiversity include tropical montane cloud forests, where the combination of high humidity, moderate temperatures, and diverse substrates supports exceptionally rich lichen communities, and temperate rainforests, where old-growth trees provide extensive and varied substrates for epiphytic lichen growth. On stone surfaces specifically, the highest lichen biodiversity is generally found in mountainous regions with a variety of rock types and a range of microclimatic conditions created by the complex topography. The conservation of lichen biodiversity has received increasing attention from ecologists and conservation biologists in recent decades, as it has become apparent that many lichen species are threatened by habitat destruction, air pollution, climate change, and other anthropogenic pressures. In Europe, several hundred lichen species are listed as threatened or endangered on national and regional red lists, and some species are legally protected under national and international conservation legislation. The conservation of lichens on stone surfaces presents particular challenges, as the stone substrates on which many rare and threatened lichen species grow are themselves subject to quarrying, construction, and environmental modification. The preservation of natural rock outcrops, ancient stone walls, and historic buildings as habitats for lichen communities has been recognized as an important component of lichen conservation strategies, and several protected areas in Europe and North America have been designated specifically to conserve important lichen habitats, including sites where rare Saxicolous species are known to occur. The ability of lichens to survive and grow in extreme environments has been a source of fascination for scientists since the earliest explorations of polar and alpine regions, and the physiological adaptations that enable lichens to tolerate conditions that would be lethal to most other organisms have been the subject of extensive research. In the Arctic and Antarctic, lichens are found growing on stone surfaces at locations where winter temperatures may fall below -40 degrees Celsius and where the growing season, during which temperatures are warm enough and moisture is available to support metabolic activity, may be limited to only a few weeks per year. Under these conditions, the annual growth rates of crustose lichens on stone surfaces may be as low as 0.01 to 0.05 millimeters per year, and individual lichen thalli with diameters of only a few centimeters may be several hundred years old. The remarkable longevity of lichens in these extreme environments has led to estimates that some of the largest thalli of Rhizocarpon geographicum in Arctic and alpine locations may be in excess of 8,000 to 10,000 years old, making them among the oldest living organisms on Earth. Although these age estimates are based on extrapolations of growth curves and are subject to considerable uncertainty. In desert environments, where moisture availability is the primary limiting factor for biological activity, lichens have evolved adaptations that enable them to exploit even the smallest amounts of available water. Some desert lichens are capable of absorbing moisture directly from atmospheric humidity without the need for liquid water, a phenomenon that has been demonstrated through laboratory experiments in which lichen thalli showed measurable rates of photosynthesis at relative humidities above approximately 80%. In the Negev Desert of Israel, crustose lichens have been found growing on the undersides of translucent stones such as flint and quartz, where they receive sufficient light for photosynthesis through the stone while being protected from the extreme desiccation and temperature fluctuations of the exposed desert surface. These endolithic and hypolithic lichen communities represent some of the most extreme examples of biological adaptation to arid conditions and have been studied as potential analogs for life that might exist on the surface of Mars or other planetary bodies with similarly harsh environmental conditions. At high altitudes, lichens on stone surfaces are exposed to intense ultraviolet radiation, extreme temperature fluctuations between day and night, high wind speeds, and reduced atmospheric pressure. And the lichen communities that persist under these conditions are among the most stress-tolerant organisms known. On the peaks of the Himalayas, crustose lichens have been recorded growing on rock surfaces at altitudes exceeding 7,000 meters, and in the Andes of South America, lichens have been found at altitudes approaching 6,000 meters. The pigmented upper cortex of many high-altitude lichens, which may contain melanin-like compounds and other ultraviolet-absorbing substances, provides protection for the photobiont against the damaging effects of the intense ultraviolet radiation that characterizes the high-altitude environment, and the ability of the microbiont to resume metabolic activity rapidly following freezing and thawing events enables these lichens to exploit the brief periods of favorable conditions that occur when sunlight warms the rock surface above freezing and moisture is available from melting snow or ice. The role of lichens in the global carbon cycle, while small compared to the contribution of vascular plants and oceanic phytoplankton, is nonetheless significant, particularly in ecosystems where lichens are the dominant primary producers. It has been estimated that the total annual carbon fixation by lichens worldwide is approximately 3.9 billion kilograms of carbon per year, which represents approximately 1 to 2% of the total net primary productivity of terrestrial ecosystems. On stone surfaces in Arctic, alpine, and arid environments, where vascular plant cover is sparse or absent, lichens may be the primary agents of carbon fixation. And the organic carbon that they produce through photosynthesis enters the local food web through the consumption of lichen tissue by invertebrates, through the decomposition of dead lichen material by microorganisms, and through the dissolution and leaching of lichen-produced organic compounds by rainfall and snowmelt. The nitrogen-fixing capacity of lichens that contain cyanobacterial photobionts represents another important ecological function. As these lichens convert atmospheric nitrogen into biologically available forms that can be utilized by other organisms in the ecosystem. On stone surfaces in nutrient-poor environments, the nitrogen fixed by cyanobacterial lichens may represent a significant input of this essential nutrient to the local ecosystem. And studies in Arctic and alpine environments have estimated that cyanobacterial lichens on rock surfaces may fix between 0.5 and 3 kilograms of nitrogen per hectare per year, an amount that, while modest by agricultural standards, is ecologically significant in environments where nitrogen availability is a primary constraint on biological productivity. The relationship between lichens and the invertebrate fauna that inhabits stone surfaces is an aspect of lichen ecology that has received increasing attention from researchers in recent decades. Numerous species of mites, springtails, saucids, and other small invertebrates feed on lichen tissue or use the lichen thallus as a habitat. And the presence of a well-developed lichen cover on a stone surface significantly increases the abundance and diversity of these invertebrate communities compared to bare stone. Studies conducted on lichen-covered stone walls in England have recorded more than 150 species of invertebrates living within and among the lichen thalli, with population densities reaching several thousand individuals per square meter. These invertebrate communities play an important role in the decomposition of dead lichen tissue and the recycling of nutrients on the stone surface, and they also contribute to the dispersal of lichen propagules through the transport of sordia and thallus fragments on their bodies. The ethnobotanical history of lichens extends back thousands of years, and various human cultures have utilized lichens growing on stone surfaces for purposes including dyeing textiles, treating medical conditions, producing perfume fixatives, and monitoring environmental quality. The lichen Richella tinctoria, which grows on coastal rocks in the Mediterranean region, was the source of the purple dye known as orchil, which was used extensively in the textile industry of medieval and early modern Europe and was sufficiently valuable to be a significant item of international trade. In traditional Scandinavian and Scottish cultures, Lichens collected from stone surfaces were used to produce a range of brown, red, and purple dyes for wool and linen. And the recipes for these dye preparations were passed down through generations of textile workers. In traditional Chinese medicine, several lichen species that grow on stone surfaces have been used for centuries to treat conditions including inflammation, infection, and digestive disorders. Although the efficacy of these treatments has not been rigorously evaluated by modern clinical standards, the future of lichen communities on stone surfaces is likely to be influenced by the ongoing changes in global climate that are projected to continue throughout the 21st century and beyond. Rising temperatures, changing precipitation patterns, increased frequency of extreme weather events, and alterations in atmospheric chemistry are all expected to affect the growth, distribution, and species composition of lichen communities on stone surfaces worldwide. In some regions, warmer temperatures and increased precipitation may lead to increased lichen growth rates and expanded distributions of certain species, while in other regions, increased drought stress, higher rates of evaporation, and increased competition from vascular plants may lead to decreased lichen cover and the local extinction of drought-sensitive species. The response of lichen communities to climate change is complicated by the very slow growth rates and long generation times of many lichen species, which mean that the effects of changing environmental conditions may not become apparent for decades or centuries after the conditions have changed. The effects of acid rain on lichen communities growing on stone surfaces have been documented extensively in regions of Europe and North America, where sulfur dioxide and nitrogen oxide emissions from industrial and vehicular sources have resulted in the deposition of acidic precipitation over wide areas. On naturally alkaline stone surfaces such as limestone and marble, acid rain can alter the surface chemistry of the stone, reducing the pH from the naturally alkaline range of 7.5 to 8.5 toward neutral or slightly acidic values. And this chemical modification of the substrate can displace calcical lichen species and favor the establishment of acidophilous species that would not normally be found on calcareous substrates. Conversely, on naturally acidic stone surfaces such as granite, the deposition of nitrogen-containing pollutants from vehicular exhaust may increase the nutrient status of the stone surface, favoring the establishment and growth of nitrophilous lichen species such as Xanthoria parietina and Fischia ascendens at the expense of oligotrophic species that are adapted to low nutrient conditions. These pollution-induced changes in lichen community composition on stone surfaces have been documented at numerous monitoring sites across Europe and have provided early warning of environmental changes that might otherwise have gone undetected for much longer periods. The phenomenon of lichen recolonization following the reduction of air pollution levels has been observed in many European and North American cities, where clean air legislation enacted in the latter half of the 20th century has resulted in significant improvements in atmospheric quality. In London, for example, where the lichen desert extended for approximately 30 kilometers from the city center during the 1950s, The implementation of the Clean Air Act of 1956 and subsequent pollution control measures led to a gradual improvement in air quality that was accompanied by the progressive recolonization of stone surfaces in the urban area by lichen species that had been absent for decades. Similar patterns of lichen recolonization have been documented in Paris, Helsinki, Munich, and many other European cities. And the rate and pattern of this recolonization have provided valuable information about the speed at which lichen communities can recover from environmental perturbation and about the minimum air quality thresholds required for the establishment of different lichen species. The study of lichen growth on stone surfaces continues to be an active area of research in the 21st century, with new insights being generated through the application of advanced analytical techniques, including stable isotope analysis, molecular phylogenetics, metagenomic sequencing, and high-resolution surface imaging. The discovery in 2016 that many lichens contain not just 2 but 3 symbiotic partners, with a previously unrecognized basidiomycete yeast fungus inhabiting the cortex of the thallus alongside the ascomycete mycobiont and the photobiont, has fundamentally altered the understanding of lichen biology and raised new questions about the nature of the symbiotic relationship and its implications for lichen growth and ecology. This discovery, which was made through the application of molecular techniques that were not available to earlier generations of lichenologists, illustrates the potential for continued advances in the understanding of these ancient and remarkable organisms. The slow and steady growth of lichens on stone surfaces, proceeding at rates measured in fractions of a millimeter per year, represents one of the most gradual and persistent biological processes observable on the surface of the Earth. A lichen thallus that is measured at a diameter of 100 millimeters on a granite boulder in an Arctic landscape may have been growing on that surface for 300 to 1,000 years, quietly and incrementally expanding its margins while generations of human beings have lived and died, civilizations have risen and fallen, and the climate of the Earth has fluctuated through warm periods and cold periods, through wet centuries and dry centuries. The lichen continues to grow, indifferent to the passage of human time, its metabolic processes activated by each episode of moisture and warmth and suspended during each period of drought and cold, accumulating biomass at a rate so slow that it is imperceptible to any observer who lacks the patience to measure the same thallus repeatedly over a span of decades. The study of these organisms and their interaction with the stone upon which they grow remains a quiet but enduring branch of the natural sciences, one that rewards patience and careful observation and that provides, through the measurement of increments so small as to be almost imaginary, a window into the deep time of the geological and biological processes that have shaped the surface of the Earth over hundreds of millions of years.